Okay. Hi, guys. So I know I'm a little bit late with this recording, and I apologize, but I do have other things to do, and I really had a rough night, so I'm not going to go into details, and it doesn't really matter. Uh, so here is basically the same lecture, the same review that I gave on Tuesday. So all exam will have two portions. One will be Scantron portion with multiple choice. It has 32 questions, if I see it correctly, and the questions that have answers that are like after E will be labeled as question 200 and so on and so on. You will have fill up in the gap, and you will have one question to define terms. No. So in terms of first portion with multiple choice, so you should definitely pay attention on processes that will cause different alleles and that are causing diversity in population. So think about what's happening at the gene level and what is causing different versions of gene to show up. Then you should be focused on the examples about E. coli and Rhodobacter cases from the first unit. So just make sure that you understand E. coli case and Rhodobacter case. That will be very important. Also, you should understand, think about the what makes one species, like the concept of species, and think about core and pan genome. Know what is core, what is pan genome. Then you need to understand all three types of carbon dioxide fixation. So know which one is the most efficient in terms of energy and which one is the least efficient. And but also you have to know which one is which one is the most distributed and which one is the least distributed. So that will be important. And also think about species that are, which groups of organism is using Calvin, which one is using Wood-Langdale, and which one is using reductive TCA cycle. Then you need to know what are possible energy sources for chemolithotrophs. So just I will give you one hint. Hydrogen is hydrogen sulfide is. So just think about what are sources of energy, and in the same way you need to know what are alternative electron acceptors other than oxygen and how is that mode of metabolism called. Then pay attention to 5 different groups of cyanobacteria. So know exactly what is the landmark for each of them and which group is coherent and which one are not. Then it's important to know about phototrophs, which one are oxygenic, which one are anoxygenic, which type of photosystems is present in one versus another, and also know what are similarities and differences in terms of photosystems between our oxygenic and non-oxygenic or phototrophs. Then you need to know very well about different strategies for that microorganisms are employing to protect nitrogenase and why is that happening. Because remember, nitrogenase for nitrogen fixation is very oxygen sensitive. So know how organisms cope with that. Then in terms of archaea, know exactly what are main organisms in all 5 phyla of archaea and what is their characteristic, like a specific landmark for each of them. Yes, what else? Yeah, then know about one very specific organism that I talked about that is very characteristic for antibiotic production. And also you have to know specific pathogenic organisms in one class of proteobacteria. So definitely you have to understand that. And make sure that you understand because when we talk about different classes of proteobacteria, so there are alpha, beta, gamma, delta, epsilon, zeta, and pathogens do persist in a few of those groups, but make sure you know which pathogens are in which group. So I will just tell you that enteric pathogens are in gamma proteobacteria, but also other proteobacterial groups like epsilon proteobacteria have different pathogens as well. So just think about that. But I will tell you that I really prefer gamma organisms. And then you need to know specificities about sulfate respiration and sulfur, so which one is better electron acceptor, and know about NO2 minus or nitrite respiration, and know about ANME, like what is the partner of ANME, most common partner. Know very well about nitrifiers, so don't get confused. Nitrification is ammonia oxidation. That's not the same like nitrogen fixation. Then you need to know differences and similarities between ammonia oxidizing bacteria and methane oxidizing bacteria, like what is similar, what is different. So that was a special lecture. You need to know about luminescent bacteria, what the name of the molecule that is secreted and that causes luminescence in luminescent bacteria, and know about the different stalks in stalk bacteria, like what is very specific for Caulobacter stalk, and what is difference when you compare it to other types of bacteria that have stalk as well. Know very well about one specific organism that I call smart neutrophile, and that is causing bad issues in our gut, hint Helicobacter, so just know about that. And know about Firmicutes. So what, like there are three types, like sporulating, non-sporulating, and lactic acid bacteria. So know which one is which. For bonus question, think about a special lecture about HAC enzyme, hydrogen oxidizing enzyme. Like what was so special about that enzyme? How that enzyme deals with the oxygen toxicity? What in its morphological structure is helping to this organism to deal with the toxicity of oxygen. In terms of written question, you need to know only one number, and that is think about when the Earth appeared, like how many billion years ago Earth behaved, and then you need to know which organisms appeared first, and which photosynthetic organism was first. And you need also to know about, you need to understand why oxygen did not accumulate even though cyanobacteria existed, like what's the reason for that. And you need to know how we called microbial fossils. So how we conclude about microbial evolution. There are two types. I mean, I won't tell you answer. And you need to know, so in terms of endosymbiotic theory, it is extremely important to look the first lecture and to know differences and similarities between three domains of life. Look the table at the last slide. And in terms of defining a concept, I mean, I can't tell you what I will ask, but I will tell you that it has to do with terms in first unit, and especially about genes, like changes at the level of DNA. And think about, I recently announced something, so because I thought that it's not well explained in lectures, so think about that. And you need to know concept of phototrophic organism, what's the difference between green and purple sulfur bacteria, and what are differences between green sulfur and non-sulfur, and purple sulfur and non-sulfur bacteria. And then you need to know about specific group of organisms that is oxidizing iron, so know how to recognize that. You need to know what is Anamox and the name of the organism that is representative of Anamox. You need to know about fermentation and how fermenters generate energy. So remember, they don't respire, they don't do etc. They're doing something else. And you need to know about fermentation of amino acids in our mouth, plaque bacteria, what's that called? So hint Strickland reaction, and that would be all. So that's all for this exam, and I hope you will do great. Best wishes, and see you tomorrow.